Hi, this is Dr. Rosario, and in this lecture video, we're going to be talking about one of my favorite topics, fermentation. This is our last topic out of chapter 9. So I love fermentation because the products of fermentation are used to make some of my favorite things. Bread, for example, like we see on this slide, or cheese, or beer, or wine, yogurt, sauerkraut. These are all things that are made using fermentation. So what we're going to be discussing in this lecture is what is fermentation, what is its purpose within the cell. So we're going to define fermentation, and then I'm going to make a very important distinction for you between fermentation and anaerobic respiration. So let's take a step back. We've been talking about cellular respiration, and in cellular respiration we have the complete oxidation of glucose to carbon dioxide along with the reduction of oxygen or other electron acceptors, right? That is our respiration. And the formula for this process is pictured here, right? You have C6H12O6 plus 6 oxygen molecules, then yields 6 carbon dioxide, 6 water molecules, and then some energy in the form of ATP and heat. If we go all the way through aerobic respiration, the amount of ATP is about 30 to 32 ATP molecules per glucose molecule. So this is the overall yield. Remember, glycolysis gives us just 2 ATP. The citric acid cycle gives us 2 ATP. And then finally, it's really the oxidative phosphorylation step that gives us the most ATP. Now, the term anaerobic means without oxygen, right? In the absence of oxygen. If oxygen is not available to act as a terminal electron acceptor in respiration, what options does a cell have? Well, there's 2 ways that a cell can still get energy from glucose without using oxygen. The first is simply anaerobic respiration. So you go through that entire series of steps. You go through glycolysis, the intermediate step, the Krebs cycle, the electron transport chain, but instead of dumping electrons on oxygen molecules, you dump those electrons on something like nitrate or carbonate, other molecules. They can act as terminal electron acceptors. Now, not all organisms can perform anaerobic respiration. Another option is to perform glycolysis and then follow that with fermentation. So as you can imagine, you don't get as much ATP per glucose for this because glycolysis only gives you 2 ATP molecules. So let's take a look back at that figure again. Now we talked about the ATP yield for each of the steps in cellular respiration, but I want to draw your attention to the top of this figure. Notice in glycolysis we get 2 reduced electron acceptors. We get 2 NADH molecules. That's not a ton, but as we move into the mitochondrion and we go through pyruvate oxidation and the citric acid cycle, we get a ton of what we call reducing potential or reduced electron acceptors. Now, those reduced electron acceptors, they have to put those electrons somewhere. They're going to carry those into the electron transport chain. If an organism cannot use an electron transport chain because it does not have a terminal electron acceptor, either oxygen or some other molecule that will sit at the end of that electron transport chain, then that intermediate step in the citric acid cycle, they do not proceed, right? If there's no terminal electron acceptor, then the only option that a cell really has is to use glycolysis, and then that small amount of NADH that is generated, the cell can get rid of that NADH by using fermentation. So fermentation has 3 main purposes. It allows for glycolysis to proceed, while regenerating NAD. We need NAD plus for glycolysis. So we can make a little bit of ATP using glycolysis, and then we can get rid of the electrons from glycolysis by dumping them on organic molecules. This is a wasteful process. Those organic molecules simply serve as electron sinks. So there's two common types of fermentation that I'm going to discuss, but really there's a ton of different types of fermentation. And the thing that they all have in common is that the molecules that receive the electrons at the end of fermentation, they are waste products. So one example is what we call lactic acid fermentation. And in lactic acid fermentation, a cell will go through glycolysis, and then it'll take that pyruvate and it'll put the electrons back onto pyruvate to generate lactate, right? Lactic acid fermentation is what happens in our muscle cells. As you're working out and your body is pumping blood and you're using as much oxygen as you can in cellular respiration to generate ATP, eventually your cells run out of oxygen, right? Your muscle cells run out of oxygen. So in order to at least keep the cells going, to keep them alive until more oxygen can be transported to the cells, they will switch temporarily to lactic acid fermentation. That gives the cells just a little bit of ATP per glucose. Fermentation is not a particularly efficient way of getting ATP. You're going to get the most ATP if you can go through respiration. Another type of fermentation is alcohol fermentation. This, of course, is going to be my favorite because this is what we use to make a lot of wonderful products like our bread and our beer and our wine. Alcohol fermentation is performed by a variety of microorganisms where they take glucose, they convert glucose to pyruvate through glycolysis, but then they use that pyruvate again as an electron sink, but here we're using different enzymes and different chemical reactions, and the products of this particular fermentation pathway are carbon dioxide and ethanol, or alcohol. So this again is another example of fermentation. The carbon dioxide and the ethanol, again, they're waste products. So think about that the next time you drink beer or wine, right? Really what you're drinking is the waste products of the microorganisms that we're feeding on the sugars in those liquids. All right, so that's fermentation in a nutshell. The thing I want you to remember about fermentation is that it does not generate ATP by itself. Glycolysis produces ATP, and then fermentation simply regenerates NAD+ so that the cells can continue to get that ATP out of glucose through glycolysis.